Good day, my name is Grant Warnick. I'm the lead research scientist at Quantum Genetix, and today I'd like to talk about genetics testing using PCR. A lot of cattle producers like to know the heredity of their herd, or whether certain animals have particular traits. Crop producers also want to know if their crops have traits, or whether there's disease on certain parts of their crops, or whether there are GMOs in their crops. And so what they do is they take samples and they submit them for genetics testing. And in discussing the results of those tests, the laboratory conducting them will often use the word PCR. And so today I'd like to take a couple minutes and just talk about what is PCR. So we'll get the initials out of the way first. It is polymerase chain reaction, PCR. And what it is basically is a copier of DNA. It just makes lots and lots of copies of DNA. It's a method that copies DNA. That's important to remember, because if DNA is actually invisible. Like we have to keep that in mind. The DNA is actually invisible, and so if you make a lot of copies of it, you can better understand the DNA. In order to discuss PCR, we should first think about the structure of DNA. Remember, it's like a ladder with a twisted ladder. So if I were to unravel that ladder and split each of those sides of the ladder in half, and so you got one long piece of DNA, single-stranded DNA. If I were to develop a few bases that were to bind on a specific part of that DNA, it will actually serve to prime that DNA. It is actually called a primer. So that binding makes it double-stranded in that one area of DNA. In the presence of DNA polymerase, the P in PCR, this DNA polymerase, if that polymerase is there, it will add the rest of the bases off of that primer. Wherever there's primed DNA, it'll become double-stranded. So again, now you have a two-sided ladder. In a second reaction of PCR, we can open up that ladder again, get our single-stranded, we add our primers, and make another double-stranded piece of DNA. So that would be two cycles. So if we do this over and over again with the polymerase and the primers, we get what is called a chain reaction. So that's the CR. It's polymerase P mediated chain reaction, polymerase chain reaction. And at the end of a typical PCR run, you will have, billions of copies of DNA. And that's really important, because if you have billions of copies of DNA, most laboratories that do genetics testing have dyes and stains and pieces of laboratory equipment that can observe these stockpiles of DNA and observe their nature. So, in the crudest sense, if a sample is positive for that DNA, you'll see lots of that stain or lots of that dye. And then if that target DNA is not there in another sample, you won't see anything. And so that's how PCR can be used to improve food production, by observing or trying to understand the DNA, trying to know if it's there or not. Now, PCR isn't the only, genetics method of testing genetic traits. There's also microarray is another one you might hear, or you might hear of restriction digests. But the power of PCR is that it is fast. It is among the most accurate techniques available, and as well as it's also cheap. It's cheaper than most other methods, and furthermore, it's customizable. It's really fast to design and carry out. So that's polymerase chain reaction. That's PCR, and that's how it helps, and that's one way it helps in food production. So thank you for watching. Please tune in again soon, where we will have more videos talking about genomics and food production. Until then.